Today we're going to talk about translation. So translation is the process of creating a polypeptide chain from a template strand of mRNA. So this is when we're taking the mRNA and we're using it to make a protein. So this is mRNA or RNA to protein is the process of translation. Yesterday we talked about transcription, which is how DNA becomes RNA. Today it's how RNA becomes a protein. So what's important to know is this occurs at the ribosomes. mRNA is read in 3 nucleotides known as codons. So it's a series of 3 letters like AUG, CGA, UUU. 3 of those nucleotides at a time is referred to as a codon. So let's talk more about what those codons do. So the genetic code is a set of rules that determines which amino acids line up in a specific sequence of mRNA. So it turns out your body knows how to read those 3 letters and turn those 3 letters into an amino acid. And that's essentially what translation is. So it's read in sets of 3 bases. Again, just to refresh, we call those codons. And there are 64 different codons for 20 different amino acids. So that gives you quite a lot of possibilities and combinations that you can have, which is how we have such diversity in our bodies when it comes to proteins. Let's take a look at one example. So let's say we have AUG. Okay, so AUG is the codon on the mRNA. And your body needs to read it. So what it does is it goes over here and it says, okay, here's the A. Okay, so A is here. And that's the first letter. Then I go to the second letter and I see a U. So then I'm going to go down, boop, 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 AU, and then going across here, G. So I have methionine, which is abbreviated with a MET. And MET is the start sequence. So this is the start. So I would say that methionine is the first amino acid. Maybe I have another one, U. Okay, so if I have U, let's try to read that one. Take a second and let's see if you can read U. So UUU is phenylalanine. So that's phenylalanine. Okay, so that's a different amino acid. So there's another one here that says stop. This one right here. And there's a couple of other stops. They're noted, denoted in the red. So if you have U, for instance, let's say you have UAA, that is a stop codon, UAA. That means everything needs to stop, and we'll talk a little bit about that in a second. But this is how your body turns codons into amino acids. It reads them, and think of this chart as like a decoder for it. Okay, so let's talk about the different steps of translation. It's really the same as transcription, just different. So the first step is initiation. And in initiation, you have this little thing here. This thing is a tRNA. Okay, and this pink thing here is an rRNA, aka a ribosome. So what happens is the mRNA binds to a ribosome. So that's that little pink thing there. And then a tRNA lines up adjacent to the start codon on the mRNA. So the AUG, we said, is on the mRNA, and we said that's a codon. Right, the opposite of that is UAC. And this is on, in this example, the tRNA, and we call that an anticodon. It is the opposite of the codon in order for it to bond there. So the tRNA has an amino acid attached to it, and it's the appropriate amino acid for the codon. So in the previous slide, we did AUG, and we learned that that was methionine, which is abbreviated with MET. And we said, oh, look, okay, MET. So that tRNA is carrying the methionine amino acid. So then what happens next is a stage called elongation. The mRNA strand continues through the ribosome and tRNA molecules attach to the correct codon. Now we saw in the last slide that it started with AUG. AUG is always the start codon. Every single sequence starts with AUG. So here's a picture. And then I'll talk more about it. So we have the first amino acid, the first tRNA with its amino acid is coming in. Then we have a second one coming in right here into this new site. So then you have these two tRNA in the ribosome right next to each other. So amino acids that are adjacent to one another form peptide bonds between each other. So when you have these two tRNAs right next to each other, they're like, we're friends, and they form this little bond. When they do that, this tRNA is going to break off and leave its amino acid behind. Then the whole thing will scooch over to allow a new tRNA to enter in. Now, I know this doesn't make a whole lot of sense looking at this right now, so I'm going to show you a bunch of animations that kind of explain how this works. So if you'll notice here in this picture, we have a chain of different shapes. This is my amino acid chain, aka my polypeptides. Okay, so I have my amino acid chain and it's attached to a tRNA. And again, this thing has just been moving along and adding one by one amino acids until they all were together. Then what happens is we get to the termination region. This process continues until a stop codon is reached in the mRNA. So there's that start, AUG, and there's three different stops, UAG, UAA, or UGA. So when this tRNA leaves, you end up with this free polypeptide here, which is the amino acid chain. And then that is free to roam. That will eventually become your protein. Everything else lets go, and you have the end of translation. So essentially what we've done is we've turned DNA into RNA, then we read in translation, we read the RNA, and we turned it into an amino acid chain. Okay, the codon signals the ribosome to release the mRNA, and then the newly formed polypeptide chain is released as well. So everything lets go. And then boom, you have it. You have a protein. And then that protein will go, or the polypeptide will go to be added onto other polypeptides and folded to become a protein.
Okay, so I'm going to show you a little animation of translation. So the first thing that would happen is the ribosome would attach here to the mRNA, and then it's going to look for the start codon, which is AUG. So I'm going to find the anticodon UAC, and I'm going to go ahead and attach that. Then what I'm going to do is have another one come in. So then the next codon is CGA. So my anticodon would be GCU, and that would attach. Then what we would do is we would have a bond formed between these two amino acids. Once that bond forms, this tRNA right here breaks off and leaves. And this whole ribosomal large and small subunit move over to the side. Then the tRNA that's up here can go and find another amino acid. So then we read the next codon, AAA, that's UUU. So now we have a different amino acid, again, the amino acid being up here. And again, I'm going to ungroup this so you can see it. So then what would happen is, again, we would have this bond form between the two of these. And then once that bond forms, this tRNA, this whole thing can scooch over. That makes this tRNA leave. And then we have another one coming in. So this is the process of translation. Eventually, what we have up here is our sequence of chains of amino acids, and that's our polypeptide.
So here's a picture that just labels everything from the animation I just tried to make for you all. So you have the rRNA, you have the codon sequence that's part of the mRNA strand, you have the amino acid chain or the polypeptide, then you have the anticodon that's attached to the tRNA, and you have the various shapes that are the amino acid.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Thanks so much for watching the video on translation, and be sure to rewind and rewatch again, especially the animations, if that helps you fully understand it. Have a great day.